Hello and welcome to our second movie on cell cycle. In this movie, we're going to deal with entry into the M and S phases of the cell cycle. And specifically, we will be dealing with the factors and events that drive these transitions. The main factors dictating progression through the cell cycle are a set of protein kinases that are activated at very specific stages of the cell cycle. Those kinases are composed of two different components. One that stays present throughout most of the cell cycle, and another component whose cellular levels fluctuate dramatically. In fact, the other component may actually completely disappear at very specific times during the cell cycle. There are very specific pairs of proteins for every checkpoint throughout the cell cycle, meaning for every specific transition during the cell cycle, there is a very specific kinase that will drive the transition into the next stage of the cell cycle. The protein kinases that drive transition through the cell cycle are known as cyclin-dependent kinases, or CDKs. The CDKs contain the active site of the kinase that is formed by the dimer between a kinase and a cyclin. For every specific CDK, there is a specific cyclin that binds to it, therefore activating it and allowing the CDK to phosphorylate a very specific set of target proteins. And it is the phosphorylation of those target proteins what drives the cell to go into the next stage of the cell cycle. It is important to emphasize that cyclins are the ones that are degraded at very specific times during the cell cycle, whereas the CDKs are the ones that remain fairly constant throughout the cell cycle. There are only 3 different Cdk-Cyclin complexes that you need to learn for the purpose of this class. The first one is the complex formed between CDK1 and cyclin B, also known as MPF. The second one is the complex formed between CDK4 and 6 and cyclin D. And the last is the complex formed between CDK2 and cyclin E. So let's start with the first complex, CDK1 and cyclin B, also known as MPF. MPF, the complex formed between cyclin B and CDK1, is the kinase that drives transition into mitosis. As such, the cyclin component that is present in MPF, that is cyclin B, is degraded right after the cell transitions into mitosis. And it is degraded thanks to the polyubiquitination of that molecule followed by the proteasomal degradation of cyclin B. And the protein that drives the polyubiquitination of cyclin B is known as APC, also known as anaphase promoting complex. Once cyclin B is degraded by that proteasomal dependent process, CDK1 is rendered inactive, and it will remain inactive until when cyclin B is synthesized once again later during the cell cycle. In fact, cyclin B is synthesized during the G2 stage of the cell cycle, and the moment that it is synthesized, it forms a complex with CDK1. Now, in order to activate this complex, CDK1 needs to be phosphorylated, and that activating phosphorylation event is mediated by the CAC kinase. CAC stands for CDK activating kinase, which is made of a complex of CDK7 and cyclin H. So CAC phosphorylates CDK1, and this is an activatory phosphorylation event because it activates the kinase. However, right after CDK1 is phosphorylated by CAC, there is another phosphorylation event that takes place, this one mediated by the Wee1 protein. And this phosphorylation event is an inhibitory phosphorylation event because it inactivates CDK1. So before CDK1 can be fully activated, the phosphorylation event that is enacted by the Wee protein has to be eliminated. And the elimination of that phosphate residue from CDK1 to fully activate CDK1 is performed by CDC25C, which is a phosphatase that is capable of eliminating the inhibitory phosphorylation that is made by Wee, therefore rendering the complex between cyclin B and CDK1 fully active. And fully active MPF has the ability to drive the cell into mitosis. But an obvious question that comes right after is, how is it possible for this kinase to drive the cell to go into mitosis? And the answer is that MPF drives the cell into mitosis thanks to the specific phosphorylation of a very well-defined set of targets in the cell. The first target that is phosphorylated by MPF, cyclin B and CDK1, are the lamins, which form the nuclear lamina. These proteins form the scaffolding structure that provides support to the nuclear envelope, and the phosphorylation of these proteins triggers the disassembly of the long polymers that allow for the formation of this scaffolding structure. So phosphorylation of the lamins is driven by this kinase, MPF. MPF phosphorylates the lamins, therefore triggering the disassembly of the nuclear lamina, which in turn triggers the disassembly of the nuclear envelope, which is one of the critical events that are absolutely required to allow the cell to go into mitosis. Another critical target phosphorylated by MPF is a protein known as condensin. Condensin normally forms a dimer that associates with chromatin, but upon phosphorylation by MPF, condensin forms multimeric structures that drive the packaging of chromatin into the fully packed DNA that is observed normally during the metaphase stage of mitosis. The formation of fully packed chromosomes is another essential requirement to allow the cell to go into mitosis. In addition to driving chromatin condensation through the phosphorylation of condensins and triggering nuclear envelope breakdown through the phosphorylation of lamins, the complex formed by cyclin B and CDK1 also has the ability to drive the fragmentation of the Golgi apparatus and spindle formation through the phosphorylation of other specific targets. And all of these are events that are absolutely required to allow the cell to go into mitosis. And with that, we have covered all the events that drive transition into mitosis, driven by CDK1 and cyclin B. Now let's look at the activity of CDK4, CDK6, and cyclin D, and how the production of these specific kinase drives the cell through the restriction point and leads to the production of CDK2, cyclin E, which then will drive the cell into the S phase. And to start with this topic, the very first important idea is the fact that stimulation of the cell with growth factors is absolutely essential to drive the production of cyclin D. So let's take a look at how this takes place. Growth factors bind to specific receptors that are located in the plasma membrane of the cell. Stimulation with growth factors leads to the dimerization of the receptors, and that dimerization leads to the cross-phosphorylation of the receptor, leading to the activation of its kinase activity, which then leads to the activation of a complex signaling cascade within the cell. The signaling cascade is mediated by continuous phosphorylation events in which kinases are activated by the addition of phosphate groups, that is, phosphorylation. Once the kinases become active, they go on to phosphorylate other kinases, therefore activating them. And a few kinases that are essential in this signaling cascade are MEK and ERK. And ERK plays a critical role in this process because it has the ability to translocate across the nuclear pore complex upon phosphorylation. And once it reaches the nucleus, ERK goes on to phosphorylate a large array of transcription factors that then trigger the synthesis of many different proteins, among others, D-type cyclins. D-type cyclins are very short-lived proteins, and therefore they are very quickly degraded. So, in order for the cell to accumulate enough cyclin D to fully activate CDK4 and CDK6, the cell must be under continuous stimulation with growth factors. Failure to achieve this continuous stimulation with growth factors leads to low levels of cyclin D. And when this happens, the cell becomes quiescent. That is, the cell leaves the cell cycle and goes into the G0 stage. In contrast, sustained stimulation with growth factors leads to high levels of cyclin D that then lead to full activation of CDK4, CDK6, allowing this kinase to go on and phosphorylate the retinoblastoma gene product, also known as the RB protein. The retinoblastoma protein, also known as RB, is a protein that is mutated in a type of cancers that affects the retina, therefore the name retinoblastoma. These cancers usually develop very early in life and are identified by the appearance of a clear area within the eye of the child affected. The retinoblastoma protein plays its role in regulating passage through the cell cycle by controlling the activity of a transcription factor known as E2F. When E2F is bound to the retinoblastoma protein, it acts as a strong transcriptional repressor. In contrast, when E2F is free of the interaction with RB, it acts as a strong transcriptional activator, driving the synthesis of key proteins that are required for DNA replication, such as DNA polymerase delta, PCNA, and cyclin E. The molecular event that dictates the release of E2F from the inhibitory activity of RB is the phosphorylation of RB by CDK4, CDK6, and cyclin D. So the kinase activity of CDK4, CDK6, regulated by cyclin D, is what mediates the release of E2F from RB, therefore allowing for the activation of the transcriptional activity of E2F, therefore leading to the synthesis of DNA polymerase delta, PCNA, and cyclin E. Mutations that inactivate RB render E2F constitutively active, and under these conditions, the cell will progress into the S phase much more easily. And this is one of the conditions that favors the development of tumors. Therefore, RB has been called a tumor suppressor because its inactivation renders the cell more prone to form a tumor. The phosphorylation of RB leads to the activation of the transcriptional activity of E2F, therefore leading to a dramatic increase in the synthesis of cyclin E. This increase in the synthesis of cyclin E leads to the full activation of Cdk-Cyclin E, and such event leads to phosphorylation of MCM. Phosphorylated MCM becomes fully active, and active MCM has the ability to unwind the DNA thanks to its helicase activity, therefore leading to the beginning of DNA replication, which is the event that determines the beginning of the S phase of the cell cycle. Therefore, the increased synthesis of cyclin E drives the cell into S phase by activating the MCM helicase. And the activation of MCM happens only once during the cell cycle, therefore providing for a restriction mechanism that allows only one cycle of DNA replication for every cell cycle. It is important to indicate that mutations that result in continuous unregulated expression of cyclin D1, such as what will happen if you activate any of these kinases in a constitutive manner, would facilitate passage through the cell cycle, therefore enhancing tumor formation. Therefore, most of the kinases involved in these signaling transduction events are referred to as oncogenes, because their constitutive activation will allow the cell easier passage through the cell cycle, therefore facilitating the formation of tumors. So, to recapitulate the transition through the restriction point and into S phase, the whole process gets started by the binding of growth factors to plasma membrane receptors, which triggers signaling cascade events leading to the activation of, among others, ERK. ERK then activates other transcription factors leading to the synthesis of cyclin D. Cyclin D binds to CDK4, CDK6 and activates them. The resulting complex then goes on to phosphorylate several substrates, including RB. Phosphorylated RB releases E2F, which then triggers the transcription of several different genes, including cyclin E, DNA polymerase, and PCNA. Cyclin E then binds to CDK2, therefore activating it and allowing it to phosphorylate the MCM helicase. Active MCM helicase opens the DNA, therefore allowing for the beginning of DNA replication, which is the event that marks the beginning of the S phase. And with this, we have finished our second movie related to progression through the cell cycle. Feel free to watch the movie as many times as you consider it necessary. Thank you for watching. And remember that knowledge is power.